Now let's take a look at another way operons are controlled. Well, there are several aspects to controlling the lac operon. The inducibility of these genes and the presence of lactose is what we will examine today. Remember, many bacterial genes are organized differently than eukaryotic genes. They have operons, or groups of genes physically close together on the chromosome that do a major metabolic function for the cell, along with their control elements. In this case, it is the three enzymes that allow the cell to use lactose as a sugar. This will be turned off either in the presence of glucose or in the absence of lactose. We'll look at the control mechanisms that occur when lactose is available. Much like the trip operon, the three genes for the enzymes involved in using lactose as a sugar are grouped together just downstream of their operator sequence. Just upstream, but separate from the operon, is a regulatory element called a repressor gene, which is always or constitutively expressed. In the case of the lac operon, the produced repressor protein is active, at least in the absence of lactose. Therefore, if glucose is present, an easier sugar to use, or if lactose is missing, the repressor protein is formed and binds to the operator sequence. This physically blocks the RNA polymerase from binding to the DNA and transcribing these genes. So if lactose is missing from the medium, no enzymes to digest lactose are produced. So when lactose is present in the growth medium, an isomer, a different chemical shape of lactose called allolactose, is also present. When allolactose binds to the repressor protein, it disengages from the operator sequence, making the operator available to RNA polymerase for transcription. As these genes are transcribed, the messenger RNAs are translated and the genes needed to use lactose as a sugar are produced. And lactose is digested, forming glucose for use in glycolysis. Therefore, the lac operon, at least in regards to the presence of lactose, is considered an inducible operon. It is inducible because it is induced to produce the enzymes that make lactose useful as a sugar when lactose is present. Unlike the trip operon, which is turned off by the presence of tryptophan, the lac operon is turned on when lactose is present. However, we haven't discussed the fact that even when lactose is present, if glucose is readily available, the expression of the lac operon is repressed. So like many things in biology, it's a little more complicated than it looks on the surface. However, what this video shows is the inducibility feature of the lac operon.